Hello viewers, welcome to Mythology. In this video, we will discuss about the quality control or the checkpoints in a cell cycle. Several cell cycle checkpoints function to regulate the cell cycle progression in response to the cell size and extracellular signals. These extracellular signals, which may include the nutrients and the growth factors. In addition, the events that take place during the different stages of the cell cycle must be coordinated with one another so that they occur in appropriate order. Cell should not start M phase before the completion of genome replication. Else, it would be a catastrophe in the process of cell division, as the daughter cells will not receive the complete copies of the genetic material. So, in most of the cells, coordination between different phases of cell cycle is dependent on the system of checkpoints and the feedback controls that prevents the entry into the next phase until the preceding events have been completed in a very orderly manner. Incomplete or damaged chromosomes are not replicated. They are never passed on to the daughter cells. The MCM proteins, they bind to the replication origins together with the ORC, that is the origin replication complex proteins, which act as linking factors that allow the replication to initiate, and thus it allows the cell cycle to start and progress in a cyclic manner. So let's see what are the different checkpoints at the different timings of a cell cycle. The first checkpoint is present towards the end of the G1 phase. In eukaryotes, that is, in the higher animals, higher than those of the bacteria. This checkpoint at G1 is referred to as the restriction point. But in the fungal or the yeast cells, this is referred to as the start point. What does it check for? Let us see that. This checkpoint at G1, it checks for any damage on the DNA. It also checks for the enzymes and other proteins along with the adenosine triphosphate, which would be required for the cell to progress if they have been produced in the required amounts or not. It also activates cyclin proteins that initiate cell division. It is called the CDK protein, that is cyclin dependent kinase, which initiates transcription. Then, if transcription has happened, then the cell enters into S phase, or we can say the cell is committed for the cell division. These are usually regulated by the ATR, ATM, P53, and MDM2 proteins. Then it comes to the second checkpoint. The second checkpoint that occurs almost at the end of G2 phase, or it is before the entry of the M phase. At this point, the check is done for the DNA replication. If it has happened properly or not. It checks for the enzymes, proteins, specifically the tubulin proteins, and for the production of the ATP if that has occurred in the appropriate amount for the rest of the cyclic process to progress. There is a third checkpoint which is referred to as the M checkpoint or the spindle assembly checkpoint. Its work is to monitor the alignment of chromosomes on the metaphasic spindle. This is mediated by the MCC protein complex, that is the mitotic checkpoint complex, which are done by the proteins BUPR1, BUB3, CDC20, MDM2 assembly proteins. This also checks for the separation of the chromosomes in the onset of anaphase. When the spindle fibers are properly attached to the kinetochore or not is also a work of the spindle assembly checkpoint. I hope you liked my video. So if you liked my video, do not forget to give it a like, share it with your friends, and if you haven't still subscribed, please subscribe to my channel. Thank you once again.